welcome to the plenary session on big data, smart data. I'm Barbara Nasto, and I'll be moderating the session today. And I'm going to borrow a page from the chemistry industry of the 1960s and dub this session Better Healthcare Through Big Data. And I'm going to say it again because I want the power of that statement to resonate. Better healthcare from big data. Now, we live in the information age now, long way from the 1960s. Big data is here, and we love it. It tells us what to buy, where to go, and it can even guide us in choosing a life partner. So I don't think there is any question that big data will impact life science, and it has the potential to greatly alter the way the world practices healthcare. So I think the real questions are, how do we best use big data to make better decisions, to optimize our resources, and cast a broader net on the world's ills? It's a pretty big objective. And what are the obstacles of making this a reality? How do we collect data? How do we manage and apply it? What questions do we address? And how do we do it wisely? How do we turn this glut of data that is available to us into smart data? So, do we need to bring the public along? I think these are obvious questions. Do we need to convince the politicians? Well, to quote one of our speakers, that is probably the first person, that is the person that's going to speak, we need to walk before we run. And I'm sorry that I stole that from you. There are a few smart things that big data is already doing, and we're going to get the global view from two very disparate perspectives on the use of big data in healthcare. Our first presenter will be Ashley Moore, who is at Woolmore. Sorry. Ashley Woolmore works for IMS Health in the real-world evidence group. Now, it's a pretty tall order. However, I'm going to let him speak for himself on what value this can bring to the life sciences and the practice of healthcare in the world. Yes, sir? Michel, I'd like to invite you up as well at this point.
Can you hear me? Yes? Thank you. I'm going to use a few slides. Before I do so, one of the reasons why I came today and was very grateful for the offer to come and speak to you, I really like the title, Big Data, Smart Data. I think where we are in this process is really this transition from being awash with data to figuring out how to use it better, and how to get somewhere smarter. And in fact, one of the reasons why I joined IMS at all was because from the outside perspective, this was a company that was leading in terms of this transformation from big data to smart data. And partly what I do for IMS Health internally is to say, okay, how do we actually get better at this? What are some of the next steps that we need to take in terms of making sure that we have access to the sorts of data and the requirements that our clients are making of us, so that they in turn can put data to work for the benefit either of the life sciences industry or the healthcare industry. So there were really just three things that we were going to talk about. So I come first. I'm the one who gets to do the definitions. So I'll just show you one slide in terms of at least what's our definition of big data in healthcare. Then I'll go on to just touch very briefly on the distinction between the ambition that we have for the use of these data and where I think we are today. And one of the observations, as Barbara was just saying, is, well, to a certain extent, are we trying to run before we can walk? My personal point of view is there are tremendous sources of value that are currently going untapped because we haven't put in place some of the infrastructure that we need to really make use of these data. And I think to a certain extent, there are some barriers to its use, and some of those are definitional. So if we get cracking in terms of real-world data sources, where we get very excited, and I'll come back to that on the next slide, is electronic data. So the world of real-world evidence and real-world data in healthcare, not particularly new. We have issues around observational studies, non-interventional studies, drug utilization studies. Where we get much more excited is when those data become electronic, because that gives us a massive uplift in terms of the scale and the rapidity at which we can use them. And part of our overall strategy, to be very simple about it, is do we genuinely have the right feedback loops in our healthcare systems that allow us to learn rapidly about healthcare interventions, the impacts of policy, the impacts of introduction of new medicines? And the answer, for me at least, at the time being, is no. So centerpiece in this, electronic data. Why are we excited about that? Well, we're moving really from a world of primary data collection, which was expensive, it was very time-consuming, it was very static, and it was relatively small scale. So in the previous session, we heard about trials of 20,000 people. Well, that's pretty much the top end. I think we're much more familiar with trials of a couple of thousand people. What we're interested in is, can we create data sets where we have millions of people, and that we can leave them switched on, and that we can get dynamic access to those data so we can see what's happening continuously with systems which have very low marginal cost and which are very large in scale. And that's where we get very excited. So the role that IMS plays, to a certain extent, is an information intermediary. And when I say, are we running before we can walk, so clients come to us and they think that it's like this. They think they can ask us for an evidence generation thing that is already built, that they can access whenever they need to, and that it's high quality, and that it's quick. And I actually think that we're here. I think we're still very much at the beginning with all of this. And the work that we do is going around hospital to hospital, signing up people, negotiating information governance, negotiating data privacy, getting access to data in a way that is ethical, acceptable to the data owners themselves, in line with information governance. And this is literally a piecemeal task. And things like, the market for data is still absolutely in its infancy. So maybe a little bit of a kind of a negative point of view, but I think there are 3 ideas I wanted to share with you about how we can navigate this transition from there being copious amounts of data to really getting access to the data that we need and that we want to put it to use. So the 3 ideas are, let's just be super clear about purpose. Let's acknowledge that there are some relatively simple rules about data, and I'll just share with you parting thoughts in terms of what are some of the existing hurdles. So hopefully you can read that slide okay. It's a 2x2 matrix, and I think what we're saying on the left-hand side here is be incredibly clear about the purpose. What is it you want to do with these data? And this comes from, relatively sort of traditional thinking around data and decision-making, 2 basic types, decision-making data and data for surveillance. And those have 2 very different implications in terms of how we need to think about the data that we need and what we can do with it. And then we have the second axis, which is, well, what is the unit of analysis? Who are we actually talking about? Are we talking about making a decision about an individual patient, or are we actually talking about making decisions around an entire population? So if we go very quickly through those 4 cells, a lot of what I hear and a lot of what people get very excited about, translational medicine, leveraging electronic medical records, clinical decision support, how do we actually make better decisions as healthcare agents to the patient that we have in front of us? I also hear people get very excited about things like wearables, surveillance to a certain extent. I think it's great, it has its place, but where we get really excited is, again, top right-hand corner. So how can we put together data sets at the level of the entire population to facilitate informed and targeted change in terms of how healthcare resources are actually being deployed today, and how can we make those changes? And I think here we're talking about tech-enabled, linked at source, comprehensive data sets that tells us about how patients are using care, not just at a single point in time, but between primary care and secondary care and tertiary care.
Ashley, can I ask you a question? What do you think we need to do to put that into place? I mean, that to me sounds like you have to get a government body to, write a check. Because I'm guessing IMSF is a private company, so, your clients are what? Pharmaceutical industry,
Yep.
Healthcare delivery agencies. Unless the questions are coming from a higher objective, isn't that always going to be a limit on what big data can do?
I think more and more, and as I was saying, I think the interesting thing is that the title of this is not just about, let's talk about big data, but let's actually recognize that there is this transition that we need to go from the abundance of data to data that actually allows us to make decisions. This notion of disentangling between surveillance data and decision-making data, again, I think is very important. So a lot of the national data sets and a lot of the national initiatives are more in the bottom right-hand corner. So treatment registries, data sets which take a long time to validate, take a long time to publish, so that you can look at national level data, except it's 18 months to 2 years out of date. It's not timely enough for us to achieve the objective of getting fast feedback into the system. So to answer your question, yes, I think there is a role for us to build things like this. So T-shaped data, which is one of the things which, as IMS, we're keen to move forwards with, is to say, there is a trade-off, and if we recognize the trade-off when we're actually doing the construction and when we're setting our objectives about building the infrastructure, we can do one of two things. So we can either say, let's be very clear that what we're trying to build national data sets for which we cover almost the entire population, but the trade-off is very thin data, so relatively clinically thin, so we don't know everything about everybody, or can we build stems, so these deep stem networks, where you make the investment, private companies, the state, collaborations, we're seeing the emergence of much more collaborative type models, where in selected geographies or using selected centers, you are making that investment in the infrastructure so that the data can be extracted, the quality of the data can be improved, that you can demonstrate to the people who are sharing their data, providing access with the data, how it's being used. They can use that for their own clinical benefit as well, and trying to create a much more synergistic relationship. So I do think, I mean, it is on the agenda. We do need to make these investments in infrastructure for this to become a reality. I'm conscious of time.
Yeah,
I'm just going to share
conscious of the time as well.
maybe one last thought with you,
Okay.
which is, well, what are some of the hurdles? So I think, particularly when we're talking about life sciences or even agencies who want access to the data, first one is lack of anticipation. So assuming that online 24 hours, always switched on portal exists, it doesn't. The market for data is currently quite immature, and not anticipating the 18 months it takes to build the relationships, to understand the data, to get it into the state that you need it to, is one of the key hurdles. Absence of scale. So we're seeing very fragmented efforts. These are big initiatives. The funding is substantial, but I think the reward of doing it is colossal. And the third one is more of a change in mindset, which is, particularly if we're talking about evidence-based medicine in life sciences, we've all gone through that evolution over the last 20 years of evidence-based medicine. The predominant mindset is of a study with a hypothesis and the data collection period, the analytical period. I think we need to embrace and move away from that, because the sorts of flows of electronic data that we're talking about allow us to move away, and I was talking to somebody last night about this, it's like moving away from photographs and moving to cinema. You're moving away from snapshots in time to something which is genuinely dynamic, and I think once we've tasted that and we've got the hang of that, we'll look back at much more static approaches and find them strange.
Do you think the idea of a real-world evidence perspective, I mean, is the digi-age going to help this in any way? Are your little devices on your iPhone? I mean, I think there's something to come later on in the session that might speak to that point. But do you see, you know, in the changing age where apps are something that have a role to play in everything we do?
Yep.
Do you see this as something
I
that's
absolutely do.
Yeah?
And so the slide I showed of the 2x2 wasn't to say that there is a better or a worse, just to say, let's just be really clear about which bit of the puzzle we're trying to address. We think that there is huge untapped value in that top right-hand corner around making changes, policy-driven around healthcare resources are used and deployed. I think the bottom left-hand corner, so surveillance of individuals, a fascinating area, will transform the relationship between patients and physicians and clinicians. So I think there is a place for all four of those pieces, but where we're sort of placing the biggest bet, if you like, is evidence-based healthcare as the next step on from evidence-based medicine. And in order to do that, we need a measurement technology that allows us to actually understand what we are doing in healthcare and to get that feedback promptly so we can make informed choices.
And how long are we away from being there?
5 years.
Yeah. Okay. So, if you don't mind, I'd like to now invite Michel Bergeron from the University of Laval to the stage. Now, Michel has a very different perspective because he has a clinician's perspective, and he's actually asked the question, can big data curtail the threat of microbial resistance? And so I now give the floor to Michel and ask you to please join me at the table.
I lost my,
Yeah.
my thing. I think I'll use a microphone instead because it fell, unless...
Well, you are coming through.
Yeah.
Well, I'll put it here. Good night, ladies and gentlemen. It's a pleasure to be here. I love Lyon, not only for food, but I love it because it's a bastion of microbiology and also of medicine. There's not a city in the world that has so many names of doctors. So I'm a physician, and I'm an infectious disease specialist. I started my practice as an infectious disease specialist in 1974. And in 1985, I woke up with a dream. How is that? Because I'm also a microbiologist. Why can't we identify microbes using, cultures and other ways in less than 2 days while we've walked on the moon, in fact, in '69? And I've been working on antibiotic resistance and also pharmacology all those years. And I've decided to move in the diagnostic field because I knew if we would use DNA, maybe we could solve the problem. I've entitled my talk From Pasteur to the Genome, the Molecular Diagnostic Revolution. During the last 30 years, especially in microbial genomics, there have been millions and trillions of data that accumulated and brought us to a new era of molecular diagnostics. A lot of big data. But in reality, what I'll show you is that when you ask a clinical question and you say you're going to use DNA, you can build up your big data for a specific purpose. And as you will see, we've used horizontal genomics instead of vertical genomics. And this has resulted in fantastic, fantastic diagnostic tools that are sold throughout the world right now. But first, I want to show you or remind you that the microbial world is big data. Microbes have survived and evolved during 4.2 billion years. Microbes represent 60% of Earth biomass. That's a lot of cells. In our gut microbiota, there are more microbes than stars in our galaxy. Remember microbiota, microbiome, resistome? This is the future of medicine. I won't talk about it today, but we're working this area also. In my mind, this is where it's going to happen in the last 100 years, because it's going to take 100 years to study this. There are 1,600 microbes that cause infectious disease, but only 100 of them really cause about 80% of the diseases. Thousands of microbes has now been sequenced. In reality, 36,000. Pathogens now have evolved extreme resistance to antibiotics. You've heard in the last few talks in the previous session that resistance is #1 problem for WHO, for Europe, for United States. Even President Obama has a special program on that. And you will see antibiotic resistance can become cured with rapid diagnostics. Infectious disease is still the first cause of mortality, about 30% of all death. And infectious disease require rapid diagnostics. You know it when you get flu, get sick. You would love to know if it's flu or other things.
Michel,
Yes.
could you speak a bit more directly to the interdisciplinary team
Yes.
that you needed to assemble
Sure. Be
to
happy
bring?
to.
I mean, I think we can stress the importance of the fact that, being able to diagnose an array of, what specific microbe is attacking somebody within an hour
Exactly.
will have a huge benefit.
Exactly.
Yeah.
So just to
But if you could tell us, because you've been at this for what, 30 years?
30 years right now.
Yeah.
And it started, I was alone as a clinician with my team working on pharmacology, toxicology. And then I decided, I had a friend I was working with resistance, Paul H. Roy, who was a fantastic basic microbiologist. And I said, I've got this dream. Could you come with me? And now we have a transdisciplinary team, as you will see at the end. We even have engineers, we have physicists, we have chemists, biologists, people from industry, but they're part of the research team, but they're no string attached. They want to be with us, okay? And so plastic people, as you will see. So this, I think, what made our success is really because we got all of these people whom I convinced that my dream should be theirs. It didn't take long to convince them, and that's what happened. So if you look here on this slide, pasture microbiology, extremely slow, 2 to several weeks, and that was the basis for but there are consequences in reality of slow diagnostics, imprecise diagnosis, empirical treatment, abusive use of antibiotics and antimicrobial resistance. They're all connected. Overuse of antibiotic and resistance, it's a use of broad spectrum, so there's a very close relation. And hospital-acquired infections are secondary to overuse of antibiotics, there's no question. Increased death, poor control epidemics, slow intervention for bioterrorist attack, and high healthcare costs. So many consequences of slow diagnostic. So I had this dream, and look at the pastor smiling when he sees DNA. I'm sure if he would live today, he would be really, really working on DNA. So that was my dream at that time, and I'm pushing it, and I'm still pushing it. Now, in '85, developing DNA-based tests posed many, many challenges. First, sample prep to extract DNA, lyse the cell. I won't talk too much about that, but that's very important. Proper molecular strategy. There were few available DNA sequences in 1985. Everybody was working on ribosomal RNA. For me as a clinician, I need to know if it's a Staphylococcus, not only the genus. I wanted to distinguish it from aureus and epidermidis, etc. So this is why we dream at that time of finding and discovering new genes that in reality will have different degree of conservation. So we could go from family to genus and species. And that's what we did. We discovered 3 genes, TUF, ATPD, and RECA, and this really was very useful. And remember, in 1985, for those who are old enough, that there were no commercially available PCR. PCR discovered 2 years before by Carey Mullis. So it's only in the 90s where PCR and then real-time PCR at the end of the 90s appear, and then practical sequencing. And I'll talk a little bit about this.
Okay. I'm going to ask you to skip to defining exactly what your diagnostic does and how it's going to change the way that clinicians practice medicine and what it's done to the University of Quebec Hospital, what you've seen.
Yeah, it's coming, but I could summarize it if you want.
Well, I just think that your slides are not as quick as you're saying, and we are running long on time. So,
Okay.
They're not very quick, actually.
Okay.
I'm
So
sorry to point
that's
that
okay.
out.
That's okay. That's okay. That's part of the debate.
Yeah.
So we've developed really genomics of cancer genes, and what you can see here, the center is phylogenetic tree, goes from top to the bottom. And on the left, on your right, no, on your left, sorry about that. On your left, you see Maurice Boissineau holding a patent, a 1,500 pages patent, which has all the primers and probes necessary to identify 450 bacteria. And this resulted, here you go, resulted in very important tests. One for group B streptococci in pregnant women that delivery prevent meningitis in kids, and then MRSA, methicillin-resistant Staph aureus, C. difficile, and other tests, which really has transformed the practice of medicine. The clinical impact of these tests, that's what you're asking, is seen on this next slide. I'm just looking at one of these tests, the methicillin-resistant Staph aureus, which was a revolution because we're the first in the world to demonstrate we could identify microbe directly from a clinical sample in less than 1 hour. In fact, half an hour to 3/4 an hour. So it reduced MRSA transmission. It reduced infections due to MRSA, surgical infections, bloodstream infection, decreased death. In one hospital during 4 years, they've used our test. 72 deaths in 4 years and 8.8 million. One test, just imagine. And it lowered the turnaround time on number of isolation days in hospital, which is very important because these bugs are very contaminated and it saves costs at the same time. And the economic impact is there. You can see I created a company, IDI, Infectious Diagnostic Incorporated, was bought by Becton Dickinson in 2006. And you see the fabrication center that they built in 2008 and the R&D center in 2007. They've invested $800 million in Quebec City. There's 350 highly qualified jobs. And finally, our products are sold in 50 countries. It's slow, huh?
So, Michel, I'd like to invite you to the table because I think there is, even though these are disparate views, come have a seat.
Yes, but I want to finish with this one.
Oh, okay.
Let me finish this one, Barbara, because
I'm so sorry.
This summarized
microfluidics is
this summarized 30 years of work. Mm-hmm. And on the first, what you see here is culture standard. So we got slow healthcare decision, more than 48 hours. In the middle, you got our tests that we have developed that are in laboratories, goes from 1 hour, 24 hours. Why? Because labs are centralized. Because they're centralized in one city or in one hospital, and the diagnostic cycle is very slow from prescription to the results. So while these tests can be done in 1 hour, the results comes back in more than 24. And what we're working on right now is the future. I call it the Nespresso of genomics. You see it on this slide. It's a microfluidic device. You can see it. Instead of having grains of coffee, we have grains of DNA. It does sample preparation. It does dilution. And you can read. It's real-time PCR with a very small apparatus. And I think this is the future of the practice of medicine. And here is the transdisciplinary team. Numbers do not talk by themselves, but to make practical sense of big data, we need this team that I've been working with for many years. Thank you for those people who've been working many years with me and believing in my vision.
Thank you, Michel. So I was wondering if each of you might comment, because I do see that there is a parallel, even though you're coming from a much different perspective than you are as the clinician, of getting the clinician to change the way they practice medicine. So I mean, would you perhaps comment on how to create a cultural shift? And I would ask you kind of a similar question as to why isn't every hospital lab doing this, you know, looking at your numbers? So first, Ashley?
So maybe just a quick reflection on the presentation that we've seen. I think that there is this necessary synergy, connection between innovation and new interventions in healthcare and our ability to figure out if they work. And I was minded by one of the slides that was shown where we were saying, okay, well, we introduced this and we reduced infections and we reduced hospitalizations and we reduced death. So I think there's this very sort of complementary thing that we're saying, which is on the one hand, this is the intervention that we introduce, and what I'm saying is let's build the management and the information technology that allows us to collect and create that evidence in as efficient and as in as seamless a way as possible. So that I'm assuming that when you introduce this into clinic, it then took you 6, 12, 18 months to collect all those other data through some sort of clinical audit about, okay, well, did we really have a clinical impact? And that's the bit that I'm really interested in, but the two must dovetail together. And I think the second part of your question is the really important one, which is scale. So how do we achieve scale and how do we really roll things out? Partly our point of view is that providing feedback to clinicians about what they are doing individually and collectively and the impact that's having on their patients individually and collectively is a missing link. And systematically and universally, every single project that we've done in this space, we start off with a relatively hostile crowd. We get them to accept the fact that the data that we're showing them are their data. And then you get into a constructive dialogue once you've got over the acceptability of the data and the analysis about getting into a change. But that's a process, and I think that the data are integral to that process as part of a change management approach. But I think the real opportunity that we all need to tackle is how do we scale things up.
Interesting. And Michel?
Well, honestly, it's, I thought, you know, that when we've put the first, had the first test approved by FDA in 2002, that was a Group B step, and 2 years later, the MRSA. I thought that the world will just phone me and say, Michel, this is the best thing after apple pie. But in reality, that's not what happened. It's a change in culture. We've realized it's really a big change in culture, without culture. It's a big shift, 150 years of microbiologist. I'm a microbiologist. I like to smell my bugs and culture. But there's a lot of, it's related to this. So the way the system works, it's the microbiology lab that decides, because the first instruments were in the lab. And interestingly enough, we sold it to BD because we could not penetrate the market with my small company that I created at that time. And so this penetration of market, you needed to push it. Today, we can say that approximately 30% of hospital labs that are, quote, university type, big hospitals, would have these technologies, but not all the tests, you know, they choose, the menu is very important. But the democratization of these tests have not arrived because they're very expensive. It takes technical skills, and you cannot do it yourself unless you know what you're talking about. And that's why we're moving in the other direction, whereby we look at the other group of labs which don't have the expertise, and that's why point-of-care DNA-based tests will be very popular. But in fact, they're selling hundreds of millions of dollars a year, but the market is 10 to 12 billion, so they've touched probably 10 or 15% of the market. And I think there's really this change of mentality. The microbiologists have to believe in this technology, and they're doing more and more, like I told you, in academic milieu. And the other milieu, they're not doing it because they don't have the skill, they don't know much about DNA.